So let's open our dependable Safari browser made by Apple.
Or Firefox.
And look at this. So what do you notice?
Guys, enough.
What do you notice about these chromosomes?
You said they're sort of fusing. Yeah, that's sort of true.
No, really close.
Really close.
No, why haven't you shown
It might help to see the next stage.
It's gonna make
What
spindle
you might
fibers.
notice that on the edge of this orange chromosome,
Is a blue part.
it's a little tough to see the other side.
the
And on the
green
blue
part.
part, on the blue chromosome, there's an orange part. So let's see what that translates into in this next stage here. Now, so this is prometaphase.
In other words, it's kind of like between prophase and metaphase. It's like right before it.
Okay. You can see it a little bit more here.
Let's keep moving. Notice the nuclear membrane on both is going away, and these two are still attached. Now on this next one, you're going to really be able to see what has happened. You see that right here? On this orange one, there's a blue tip, and on the blue one, there's a what tip, Sarah? There's an orange tip. These chromosomes have literally switched genetic information. So now, let's say the gene for hair color is on this spot. Well, the dad's gene was for blonde hair. The mom's gene was for red hair. Now, this chromosome that has mostly the genes from the dad now has the mom's gene for hair. So it has. So then, what you have is a lot more variety. But this one, the one on this side, does this dad's chromosome have the mom's gene for hair?
No.
No.
No. So can you see how once these guys split in, well, it's going to be anaphase 2, you're going to have some good variety between all these chromosomes. Not one of these chromosomes is going to be the same. How about over here? This was the mom's chromosome. This is the dad's chromosome, but this isn't a body cell, one that carries out mitosis. What do you notice about the, are they different here? Okay, yeah, that's one difference. They're straddling the equator. What else about the genetic information on these guys? As far as, does the dad's chromosome have genetic information from the mom on mitosis?
No.
Nope. So that's kind of a cool thing. One of the differences. So there are two differences on this phase. Here, this is what phase? Metaphase, because it's in the middle. These guys are on the middle. In mitosis, they straddle. In meiosis or metaphase 1, these guys do not straddle. They just go across from each other, across from their homolog pair. Do you remember that? Homolog is, how do we describe homologs again?
Huge. Big. Big and the same.
Yeah,
Same in size.
they're the same size. Why does this iPad not like? It's weird. Okay. So that's metaphase. The main difference is this straddles, this doesn't in metaphase 1. And also, these have crossed over, we're going to call it today. All right. Next is anaphase, because they're going apart in anaphase, apart. And they're moving apart. So it looks fairly similar here, except right here, what do we notice about, do you guys remember the centrosomes? What a centrosome is? It's the, I guess I should have let that in a little better. It's the spot where these chromosomes are connected, the sister chromatids are connected, okay? So, are the centromeres together right here? No, they've been split in mitosis. How about here? Yes, they're still together. All right, let's keep going. Telophase, they are far apart. Teleport, telephone, whatever.
Phase.
Telophase.
Yeah, telophase means the far apart phase. They're far apart from each other. They're on opposite ends of the cell. Nuclear membranes forming in both, like we've learned before, the opposite of prophase, right? Then we now have, here, we have two cells in mitosis. How do they compare to each other? Kelly? They're pretty identical because those things straddled the middle, so one identical side went this way, one identical side went the other way. They are now identical. Why do I have to do that again? Well, anyway. Right. So now mitosis is done. It has two identical daughter cells. Meiosis. How many steps are left in meiosis?
7.
7 steps left-ish. Okay? It's about half done, whereas mitosis is done. Because this, would you describe this as haploid or diploid, these cells here?
Haploid.
Diploid.
Haploid.
Diploid.
Who thinks they're diploid? Haploid? Ooh, about half and half. They're diploid. Both of these cells are diploid still because they have 2 full sets of DNA. Now we're going to get this into haploid so it can become a sex cell. This is not a sex cell yet. Here comes meiosis 2. We call this set 2 of meiosis. So now we're going to see interphase 2, where the chromosomes come together. Prophase 2. That chromatin is now packed into chromosomes again. Prometaphase, that's kind of in between stage. We won't hit that too much. But you can see they're starting to come toward the middle again in metaphase. Now take a look at this. This time, in the metaphase 2, are they straddling the middle or across?
Across. No, straddle.
They're straddling. So it's different from the metaphase 1. So this time, how's this one going to compare? Well, if these things didn't cross over, how would this compare to this? If they didn't cross over, what's that, Emily? They'd be identical again. But what's the whole point of meiosis again? To be different, have variety. So that's why they cross over. So these guys cross over, so then they're a little bit different. Then anaphase again, telophase again, and then we have now split the cell. That process is called cytokinesis. Okay, and now in mitosis we have 2 daughter cells that are the same, and 4 daughter cells that are not the same. And are these diploid or haploid? Haploid. They have only 1 full set of DNA. Whereas this has 2, a mom and a dad set. Okay?
This is silly. Right. We're gonna.
Now we are going to finish what we talked about yesterday. And then hopefully we'll get into more of the meiosis differences and stuff. We're going to just draw a few of those steps. Do we want to draw all 15 steps of meiosis?
Yeah.
Probably not. I want to hit the main ones though, okay? Alright, so let's talk about prophase. The basics of, we should say, of correct meiosis. Okay? Because I want to write correct in there because I don't want you to think that this is the basics of how Down syndrome happens, okay? So we want to draw the basics of correct meiosis. Let's say there are 4 sets. We have a big set of that, and a little set. Wow, that was too big. I want them to be about the same size as this. So this is early prophase, but then this is early prophase. Now, if I'm going to talk about I'm going to show crossing over a little bit. So let's draw the blue big one again. Except right here we're going to do a little switcheroo here, because the blue one is going to get a red tail, and the red one, which we'll draw here too, it's going to get a blue tail. If you want, you're like, oh, I don't have colors. You could do this option. You can make an empty looking one with that kind, and a dark one with a light tail. Accomplishes the same thing. Does that make sense? I'm going to keep going with the color though. Alright, so we have the big ones. The same thing might happen with the little ones. So, oh, I didn't. So then this gets a little red. Actually, I'm going to cross them over in a different location, because I don't want you to think that it crosses over in the exact same spot. So I'm going to move that over here. And so we could consider this prophase, okay? Prophase 1 of meiosis. This is early. I guess this could both be considered early, because in prophase they you could say both of them are. Because remember, prophase, it's not a certain point in the life of the cell. It's like a process, okay? In prophase 1, the chromatin packs into chromosomes and they start crossing over, okay? So this is both, I guess I could, this is both prophase 1. And then we'll do metaphase 1. And then we're going to skip anaphase, telophase, interphase, and prophase 1 again, because that's kind of what we can kind of assume what's going to happen there. But we'll actually write the words at least. So now we're going to have metaphase happen. So now we have the equator, right? So this guy goes across from this guy. And then I'm going to draw the other red one over here. Sorry, Jordan, that I'm covering it up here. Did you notice how I arranged this? Are both dad chromosomes on one side? No. Could they be on both sides, on that side? They could. But I just put them in opposite so you don't think that all the dads have to be on one side, all the moms have to be on the other. You can have tons of combinations, which actually makes the odds of a mom's egg and a dad's sperm coming together and making the same identical genetic child basically impossible. Think about that. These processes, this whole meiosis thing, allows the gametes to come together, and basically it's almost impossible for them to produce the same genetic child, because there's crossing over, there's mismatching. Remember, there are 23 sets of these, 23 of these. If this is one, so there are 23 sets of these guys instead of 2. Right now we're showing 2, so we don't have to draw them all. Okay? But there are 23 sets of these. Now, so imagine, maybe there are 22 sets of these where the dad is on this side and 1 where the mom's on this side. Or maybe 17 and 6. Or, that sort of thing. There's so many different combinations. Plus with the crossing over, it's impossible for us to have the same genetic material.
Do you mean the same, two children that look the same, or the child looks the same as the parents?
The children that look exactly the same.
So about not as twins, but as born at different times.
Either way. Now, there's one case called identical. Has anybody heard of identical twins? Yes. Okay, there's seven in Gothenburg. Right. Because they're genetically the exact same. But the reason is a little different. What happened with them is they, one zygote splits into 2 zygotes that are identical. That zygote goes into which one? Mitosis or meiosis?
Mitosis.
Mitosis produces 2 cells that are exactly the same, and instead of them growing together and that sort of thing, they completely split off and become 2 separate zygotes that then.
So what's the case for non-fraternal twins?
Non
Fraternal twins.
fraternal twins, then 2 eggs from the mom got fertilized rather than just 1. 2 eggs came down the fallopian tubes, and there are 2 separate sperm that found those eggs, and then formed 2 separate zygotes.
What happened with?
And that's why you guys have you look different, because you were a separate egg, sounds weird, right? You were a separate egg and a separate sperm that came together, and you were a separate egg and sperm that came together with different genetic material because these things lined up differently in this stage. Okay? This stage is the crux of why we are different, why everyone is different.
Wait, you know what? You heard about that?
Funny word.
You heard about that girl who had octuplets? And they were all looked alike. How would that happen?
They can look alike, sure. You know, you can have certain combinations that are fairly similar.
They all look the exact same. They're all identical.
What about
It's weird when
Part of me
people
doubts it that they're completely identical. Show me the article. Like, now, is it if it's possible for one zygote to split into two, is it possible for those two to split into four? Four identicals? Maybe. I guess. I've never heard of it happening, but I guess, maybe. So that would maybe be, if they are identical, that's what I would say happened. Other questions? Are we recording right now?
Yeah, what about conjoined twins?
Conjoined twins? Yeah. A lot of times, from my understanding, what happens with those is somehow they kind of got meshed together a little bit, and they never got to separate. But usually those twins are like, they are different genetic material from what I've seen, aren't they? Sometimes you have them like where they're the same child, like they're the same zygote, and the zygote doesn't completely pinch off from itself. So then you have two zygotes sort of acting separately, but they were attached, so now they would have to be surgically separated. So in one case, if they're identical twins that are separated, then that means that they probably didn't split completely off when a normal zygote would. With identical twins, they completely pinch off. With identical Siamese twins that are attached, then they didn't completely pinch off, and now they're developing side by side. If they are twins like Jordan and Josie that say they were attached, like they're different but they're attached, it's probably that somehow they got pushed together or something and didn't quite separate. I don't know. That's my assumption.
Mr. Johnson, please call 101. Mr. Johnson?
I haven't gone in depth in studying that, so what I'm telling you is just my conclusions off of what I know about how things work, okay? So I could be wrong in areas, but I think I'm right. Let's say that. Okay, all right. So these guys are lining up across from each other in the middle. Again, there are 23 of these pairs right here, okay? You don't have to necessarily make those bracket things. But there are 23 of these pairs that can flip around. Maybe 18 dads go on this side and 5 moms go on the other. And they're different every time. So anyway, this is Meiosis 1. Now let's skip. We know what happens in anaphase and telophase.
You mean metaphase one?
Yes. Metaphase 1. Thank you. I tell you what, I'm really glad that you guys are not afraid to, that you are paying attention. Yes.
Nick was.
I'm glad you're paying attention and that you're bold enough to know, hey, this can't be right. So good. Metaphase 1. Now let's draw an arrow where we have anaphase 1, telophase 1. What's after telophase then? Interphase 2. And we're going to do prophase 2 as well. So I'm doing that, I'm drawing these so that I'm writing these so that we don't have to draw them. I think you know what happens. In anaphase, they go apart. In telophase, they're separate and the nuclear membrane comes back and they form chromatin. Then in prophase, they go back into chromosomes. Whoop. So after prophase 2 comes what? Oh, by the way, don't regard that. In other words, disregard what I just drew. Because when after telophase and interphase, we have 2 separate cells, right?
Yes, sir.
So now we have this one and this one. And these have, the ones from this half go to this one, and the ones from the bottom half kind of go over here. So now we are going to, sorry this is a little bit tedious, but I think it's interesting. Now we are going to separate them this way, and now we're going to have the equator be along this line. So we're going to have this blue, the big blue one, still with its red tag on the end with the mom's hair color. We'll just say it's hair color, okay? The mom's hair color. Along with the, looks like we have this guy too. And then down here, looks like we have, the small one is the blue one, so. And it doesn't really matter where they are, so I'm just gonna draw the blue with the red.
Bless you, Jade.
And keep in, which phase is this again? How do you know?
Metaphase.
It's in the middle, yeah. And this time it's straddling the middle. By the way, it might be helpful for you to know, how does Down syndrome happen?
Like this.
You have, which one is extra?
Metaphase?
The 21st one. Tri means
Oh, I
what?
thought that was her name.
Tri means 3 chromosome, 3 chromosomes. The 21st has 3 chromosomes. The child has 3 chromosomes. Well, how do we get 3? Here's how it works. In metaphase, instead of these guys splitting off, both pairs would go this way. Like when anaphase happens, like both of these pairs, say this is the 21st chromosome. Both of these would go to this cell rather than going across from each other and splitting off. So now when that happens, actually, hold on. It's probably more likely that it's right here. So instead of splitting off this way, one set goes completely this way. So now this cell, this side, when this splits off, this would have 2 chromosomes in that gamete, and then when it meets with another, like let's say this is describing a male. When this one meets with a female egg, these 2 are gonna be a part of the same cell as the one from the other. Like when, so say this is separates to be a sperm cell. Let's just draw it out. That'll be easier than me just saying, oh, this and this. So now it's going to turn into 4 separate cells. By the way, anaphase happened, anaphase 2, and telophase 2, happen in between here. Now we have one big blue chromosome and one red with a little tag. And over here, we have a full red one and a blue one with a little tag, the big blue one. And it looks like here, we now have the small blue one and a big red one.
What is doing?
Are you guys confused?
Ever so slightly. Why did this?
Yeah, ask me questions. What's that?
Why are those really like awkward little worm-looking things? Like, why are they awkward?
Because they split off right here. When guys, please, guys, stop.
Thank you. It shows me the division because there's like 4 pieces and only 2.
I'm getting there. So big one, small one.
Yeah. I never texted any of you.
Okay, so can you see what's going on here? This is the basics of this is the end product. Except there would be 23 of these little guys. Where there's just 2, there's actually 23. But in humans, what's that? In each of the end ones, there would be 23 for humans. Now,
And you mean 23 pairs, right?
actually, no, there are 23 just single ones in the end one. In mitosis, there would be 23 pairs because it makes diploid, so there's 2 pairs of everything. But this one's purpose is so these guys are either in the form of eggs in females or sperm in males. Okay? And each one of us is going to only have 1 full set of chromosomes. So you can see each one of these cells is different from each other. And all right, so let's go back to the trisomy 21, the Down syndrome. Let's say this, normally it should split off and make these, but in Down syndrome, maybe this whole thing goes to this side. So now this cell would have both chromosomes of that over here. And if this one were to join up, if this cell were to join up with say a mom's egg, that mom's egg also has that, let's say that's the 3rd chromosome. Let's say the mom's egg also has a 3rd chromosome, and there are 2 3rd chromosomes in this one. That makes a total of 3.
So it's pretty rare to have that though.
It's pretty rare, and it probably happens with other chromosomes, but it's simple. Maybe if a sperm and egg come together and there are 3 of another type, it'll kill the egg. It'll kill the zygote, and the parents don't even know, you know? So that's the basics of meiosis.
Thank you.